Welcome back, class, to what is probably one of the most terrifying chapters of the semester. It is all the ways that a pathogen can hurt you. So we're going to talk about a bunch of different things that bacteria and viruses and other pathogens are able to do to be able to grow and cause infection. But when they're doing this, I don't want to pretend like bacteria have any sort of conscious brain activity going on. They're not trying to cause infection. They're not being bad guys. They're just growing where they can. And sometimes when they grow, they make things that end up hurting us. So I don't want to paint this as bacteria being evil. I just want to paint this as evolution in action, where sometimes the bacteria are able to do things that harm us, and sometimes we'll come back and develop ways that our bodies can protect ourselves from it. So some terminology to begin with, pathogenicity and virulence. These are a couple of terms you should be comfortable with at this point. ID50, though, this is a new term. This is the infectious dose for 50% of a sample population. That means it takes this many numbers of bacteria to make a person sick, or it takes this many particles, this many viral particles to make a person sick. So how many particles does it take for half of a sample population? There's going to be some variation, people who are more or less tolerant of that infectious disease. But half of the population, what that number is. So it tells you about how virulent a microbe is. LD50 is a lethal dose for 50% of a sample population. So this is when we're talking about toxins, how much of a toxin half of the population would have to consume or be exposed to have a lethal dose, to end up being killed by that dose. It measures the potency of a toxin. The first thing a microbe needs to do, if it is going to make a person sick, is it has to get inside of the person. So it is going to have some sort of portal of entry. This could include mucous membranes. Mucous membranes line your respiratory tract, so it could be something you breathe in. They line your GI tract, it could be something you eat. They line your genitourinary tract and your conjunctiva, or your eyes. It might get through intact skin. So integument, or skin, would be going through a hair follicle, going through a sweat gland, or just boring straight through the skin. It might go through a parenteral route, which is going through broken skin. So it's deposited into the tissues underneath the skin. It doesn't have to bore through it. Something like an injection, if you're using a dirty needle, or if your patient is using a dirty needle, they're more likely to get a disease. Punctures or bites, cuts, wounds, and surgery. And most diseases have a preferred portal of entry. So how the disease is going to show up, how pathogenic it is going to depend on what portal of entry it used. Anthrax is a good one. It's different in terms of severity if it is on your skin or if it has been inhaled. I also think of variolation. Variolation is what was done in ancient China as a way to protect against disease. If you were exposed to smallpox in the wild, just naturally, it had about a 50% chance of killing you. But you could take smallpox and rub it into the skin, and it only had about a 10% chance of killing you. So it was much safer to be exposed to smallpox through the skin than it was to be exposed to smallpox through your respiratory tract, because that portal of entry makes a difference in how severe that infection is going to be. Then when the microbe is in your body, if you just flush it away, it's not going to cause disease. So adherence is required. That means the microbe has to attach to some structure, attached to your cells or something else. They may form biofilms, but this is required for a microbe to be pathogenic. Many microbes have adhesins on them. Adhesins are ligands, or things that can stick. Adhesins or ligands are proteins on the prokaryotic cell that will bind to carbohydrates, something like mannose or other sugars, on the surface of our cells. Adhesins in prokaryotes include the glycocalyx, fimbriae, and M protein. And our cells, people cells, have receptors. Receptors are the complementary surfaces that can bind to adhesins. So if we look at the picture, here's our cell with these purple receptors on it. And here's a pathogen, which has these little teal colored adhesins. And you can see the adhesin just binds right to the receptor, allowing that pathogen to adhere to the cell. This happens in E. coli here, which is growing on bladder cells. And this happens in staphylococcus, which is here, the purple cells, growing on the skin. So you can list those 3 portals of entry and describe how a microbe could gain access through each one of those. Then here's a little thought question, a little bit of math. The LD50 of botulinum toxin, do you remember what LD50 means? Go back and check. The LD50 of botulinum toxin is 0.03 nanograms per kilogram, and the LD50 of salmonella toxin is 12 milligrams per kilogram. So stop and remember, which is bigger, a milligram or a kilogram? Which one is more potent? Which one does it take a lot to make you sick? And which does it take only a little bit to make you sick? And how would a drug that binds mannose on human cells affect a pathogenic bacterium? If the drug is binding to mannose, what would happen to a bacteria? Then the bacteria has to be able to get past your immune system. So we have white blood cells going around ready to phagocytose or chomp up and eat bacteria. We have T cells and B cells going around looking for specific pathogens that are just looking to kill any infectious agents. So somehow these bacteria have to avoid your immune system. Some bacteria produce capsules, which can prevent phagocytosis. The exact mechanism isn't known very well how this happens, but capsules are probably either involved in just kind of hiding the bacteria, covering up its receptors, or helping to just make a slippery surface that makes it more difficult for a white blood cell to phagocytose it. Capsules entirely decide if Streptococcus pneumoniae is pathogenic or not. That is the big deciding factor for Streptococcus. Haemophilus influenzae and Bacillus also are able to make capsules. Some bacteria have cell wall components that also help them get past your immune system. The M protein in Streptococcus pyogenes helps resist phagocytosis. The OPA protein in Neisseria gonorrhea helps it inhibit helper T cells, which we will talk about in chapter 17. And mycolic acid, or that waxy lipid on the surface of acid-fast cells, helps resist digestion. We see this in Mycobacteria tuberculosis. Then some bacteria produce enzymes that they can secrete that have an effect on the area around them. Because they are secreted, they're sometimes called exoenzymes or extracellular enzymes. This would include coagulases. Coagulases are enzymes that clot fibrinogen, so that allows the bacteria to grow in an isolated area protected from the immune system or other defenses of the host. Some bacteria will produce kinases. Kinases work the opposite of what collagen is, so rather than hiding themselves within a clot, they break down the clot and allow that infection to spread. So if your immune system is kind of rounding up an infection and covering it up in a clot to kind of keep it away from the body, a bacteria that produces a kinase would be able to spread. Hyaluronidase is an enzyme created by some bacteria that can hydrolyze or break down hyaluronic acid. This is a sugar that helps hold together your cells to form connective tissue. So by breaking that down, those cells aren't held together quite so well, and the microbe is able to spread away from the infection. And collagenase, which breaks down collagen and also allows the microbe to spread. Then some microbes can do antigenic variation, which I think is fascinating. So if you were thinking about like some guy robs a bank and his face is on the news, have you seen this guy? We're looking for him. That guy is probably going to cut his hair, dye his hair, change his clothes, do something to make himself look different. So as he's going around town, people don't recognize him. Microbes do something similar when they are performing antigenic variation. So when a microbe undergoes antigenic variation, they change their surface antigens. They change the sugars and the proteins on the outside of their cell so that any antibodies that the body has made are no longer able to bind to that pathogen. Microbes are able to undergo antigenic variation by activating alternative genes, which just creates new proteins that get sent to the plasma membrane. Influenza virus is a great example of this. You have to have a new influenza vaccine every year because it has mutated the virus enough that last year's vaccine won't work for this year's outbreak. Neisseria gonorrhea is also able to undergo antigenic variation. And Trypanosoma brucei gambians, which causes African sleeping sickness. So if we look at the trypanosome, this is a type of parasite. And so if it gets into your body and starts causing an infection, it's going to start growing and growing and growing. And eventually your immune system is able to say, I recognize this guy, and start working and fighting and killing it. So it'll start getting destroyed. But right about the time that your immune system starts killing this clone, it'll activate new proteins. So now it looks totally different. And this second clone is going to start growing. And eventually your immune system is going to catch up, start making a response and fighting the infection. So that clone starts getting killed and it activates a new set of things. Clone C. And eventually it's growing. Your immune system catches up, starts killing it, fighting that infection, and it activates a new clone. It can go on for something like 10 years in some people, where it just keeps activating alternative genes, changing how it appears, until it's finally gone through all of its genetic combinations and your immune system is able to fight it. Then, at this point, the cells have gotten into your body. They have gotten past your immune system, but they've not actually done anything yet. The next thing they have to do is get inside of a cell. They can use things called invasins. Invasins are proteins that bacteria make that can rearrange actin. Actin filaments are what make the cytoskeleton of a cell. And just like your bones, your skeleton give you the shape of your body. The cytoskeleton is what gives shape to a host cell. And if that cytoskeleton is rearranged, that means that the cell is going to have a new shape. One of the shapes that it can form is it can form ruffles. Membrane ruffling is when the cytoskeleton has been disrupted to the point that it just creates a little ruffle that the microbe sinks right into. It just gets right into the cell to that ruffle. Then once it's inside the cell, the microbe can use the actin to just move itself throughout the cell, throughout the cytoplasm, and then it can jump from one cell to another by using that actin. So here we have, this is probably the digestive tract, like your intestines or somewhere. All of these projections that your cells make to help absorb nutrients from the food you're eating. But this bacteria, the Salmonella typhimurium, has altered the cytoskeleton in this area. You can see it doesn't have the same morphology. It's now got these ruffles. Here's another bacteria that's made another set of ruffles. And the bacteria just sinks right into that ruffle, gets right into the cell, and it can spread through the cell, spread from one cell to the next. And because it's inside of a cell, it's not necessarily so well presented to your immune system, and it's able to spread and cause infection. So at this point, you can tell me, what do capsules do? What do M proteins do? What functions do they have in common? Would you expect a bacteria to make coagulase and kinase simultaneously? What are each of those exoenzymes doing? How would they affect a bacteria? Many vaccines provide years of protection against a disease. Why doesn't the influenza vaccine offer more than a few months of protection? And how does a bacteria cause membrane ruffling? Okay, I'll meet you back in our next video where we'll start talking about actually making people sick. See you in a little bit. Bye.